Welcome to Scigest, your fortnightly serving of digestible science from plant and food research.
Kia ora koutou listeners, welcome to this episode of Scigest. I am your host, Jay Jayaraman. In today's episode, we're talking about clustered, regularly interspaced, short palindromic repeats, a mouthful that's abbreviated to CRISPR, a topic that's in the zeitgeist of current science comms around the world, and garnered the 2020 Nobel Prize in Chemistry. Today's guest is Zac Hanley, a General Manager for New Cultivar Innovation, based here in Tamaki Makaurau, Auckland. Welcome, Zac.
Kia ora tātou.
So, Zac, how's your position linked to gene editing?
So I'm the General Manager for New Cultivar Innovation, which is our collection of maybe 170 or so scientists who are all trying to create new varieties. And that means that's the breeders, but it's also the molecular laboratory people, the geneticists, the field folks, the data scientists, all of those who are trying to create tools so that those breeders can do their job better. And I'm here to talk about gene editing, CRISPR, because that's one of possibly the most exciting tools that's come out in the last 10 years or so.
Great. So what is gene editing?
Well, it's a complex molecular technique that does something really simple. We have to do quite a lot of work to be able to create new varieties. We need to do a lot of crossing and mixing and intermingling. And one of the things that has interested scientists for years has been, can we get inside cells and make tiny little changes that might make the job easier or might make us more efficient, more effective, more able to provide what it is that we're trying to provide to achieve the goals of what we're here for. And CRISPR, gene editing, is a kind of neat little set of molecular scissors that can go in and make a tiny change, the tiniest possible change. And if that change is meaningful, if that change has a consequence, if it makes for a better variety, a better crop, then we'll have succeeded in our goal by using this neat little tool that will get us where we need to be faster.
Okay. So is this something that scientists are using now? Are there any gene-edited plants available to buy right now?
There aren't gene-edited plants available to buy here in New Zealand, for example, but scientists all around the world are working on gene editing. It's been successfully shown to work in plants for a decade or so, and already there are a few crops out there in the world that people are either trialing or developing, or even a few of them, some tomatoes, some salad greens in the USA, that are available in gene-edited forms that have been tweaked ever so slightly. The salad greens have been changed so that a bitter taste has been taken away. Some of the work in tomatoes has been to provide healthy compounds in there, maybe vitamin D, things like that. So a few of them are starting to emerge, but largely they're within single countries. They're not really around the world because this is still a complicated and changing landscape. This is a new technology and people are trying to understand how it can be used, but also how it should be used.
So what are some barriers then to using it, just diving in and just starting some gene editing?
Well, it doesn't work the same way everywhere. There's a simple drawing that you can find on Wikipedia of how gene editing works, and that's a nice schematic that then has to be turned into something that will work in a crop of interest. If one of the scientists wants to explore the use of the tool to make a difference in one crop, then you need to kind of translate the machinery into that crop. You then want to move on and do the same thing in a different crop. You need to translate it again into the language that that new crop speaks. That kind of customization takes a lot of effort, a lot of finesse, a lot of work, and it means that you've really got to be committed to what you want to do before you undertake that. We have the kind of people who are working on that. Quite a lot of our scientists increasingly interested in whether we can achieve some of our goals using it, but you got to do a lot of work.
Right. So it sounds like it doesn't always work. Why do you think that's the case?
Well, in some cases, it's because there is quite a lot of understanding of the basic machinery of a plant cell needs to be in place for you to do it. You need to be able to work with that species. You need to understand its genome, all of the DNA that tells it how to be what it is. You need to understand the traits which you're trying to change. Maybe you're trying to change the color of something or make it fight off a pest or a disease or to grow in a way that's more amenable for us to harvest it. Any of those sorts of changes, if you don't understand the basis of it, then you can't find the single small change you need to do in order to make an improvement. Without enough people working on it, that means that those big problems are really difficult to tackle. If there's only a few people trying to solve that puzzle, then the time it's going to take you to get to an answer where you can bring in your molecular scissors is going to be a long time. In some other crops, there are 1,000 scientists around the world working on it, wheat or cotton, 10,000 scientists maybe. There's a lot of work, a lot of knowledge gathering, and you need knowledge. It's a really knowledge-hungry piece of science.
Right. That's because the background needs to be understood the high level of depth?
That's correct, yes. What we're trying to do is to understand how to make the smallest difference with the biggest effect. And until you can do that, until you understand just what your genes do, how they interact with each other, what teeny tiny change you can make, and how you can get those molecular scissors inside the plant cell in order to make a tiny difference, and then get back out and turn that plant cell into a plant and test it to see whether it's worked or not. That's a lot of technical challenge, and it can mean that you could spend many years trying to achieve something which, on the face of it, the breeder outdoors doing a breeding program might be in a race with you and might achieve much the same thing if it takes you too long.
Right. So plant biology is a much older field than, let's say, gene editing and CRISPR. So it sounds like a lot of the really fundamental stuff is what you're talking about, this sort of knowledge growth to understand what the plant biology looks like before you can go in to make those modifications using CRISPR or gene editing. So why is that the case? Even though plant biology is so old, why can't we just take those lessons that we've learned from, what we call our model plants, like Arabidopsis thaliana or Nicotiana benthamiana, which is an Australian tobacco? Why don't we take those lessons and just deploy those lessons in a plant that we're interested in?
Those things can teach us a lot. They can certainly teach us and give us all the clues we need to shorten the quest to the answer within the crop of interest. But if you want to make a change in, a tomato, then what you've learned in tobacco or Arabidopsis will only take you so far. And certainly if you're looking for what would be, say, for example, early harvesting, say, or increasing the vitamin content of fruit, then there's not a lot you can learn in Arabidopsis that will really take you all the way there in tomatoes. You've got to start to do your work in tomatoes. If you're looking to apply it in another crop or another fruit or another vegetable, then again, there'll be some specifics about that. What happens in a potato is not going to be the same as what happens in an apple. And you need to learn the specifics. Having got the general principles and understood the basis of a trait like vitamin D or red color, that will give us some clues on where to look in an apple or a potato or anything else that we want to make a change in. But that won't give us all the answers, and we need to do that work too. That said, that body of knowledge that you accumulated over many years in many different crops is a lot of the clues that we need. We've in effect been working for decades and decades on building up the body of knowledge that makes this go faster. If you were starting today, you'd have a long way to go. So we can take the knowledge of the science community, knowledge of our own research, bring that together and put those behind the molecular scissors.
Okay, so it sounds like there's a lot of work behind the biology, understanding how these organisms work. So what about the technical side? If we wanted to, let's say, do some gene editing in a specific plant, how successful is it if you just wanted to dive in and edit its genome?
It can be pretty challenging. It makes a difference which plant it is. And by that, I don't mean that some are easier than others. They are, but some days are easier than others as well. It's quite a challenging piece of work to be successful. You might have to do quite large numbers of experiments in order to have one or two successes. Maybe 20% of the time you will be successful, maybe less. You'd also have the challenge of when you're going in there with the molecular scissors and making a few cuts, or usually one specific precise cut that you want to make, then what we're doing in this mechanism is making that tiny cut and then expecting the cell, as it does every day, every moment of every day, to look after itself and to fix that cut. And we're looking for those rare occasions when it makes a mistake and it makes a tiny change. And that tiny change is the change that we've been seeking. Most of the time it gets it right, in which case the change has been made and then unmade. and you're back at square one and you haven't achieved what you set out to do. So that, again, makes it a little bit harder to achieve. But in a practical sense, one of the most difficult things is that whenever a scientist is working in a laboratory situation with a plant, you're usually dealing with sterile plants, partial plants, individual cells, and having knocked those around a bit in the laboratory, then to be able to bring them into an environment where they can start to grow up and become a fully behaved ordinary plant is really tricky. There's a lot of subtle techniques and getting the right soup for them to swim around in, getting the right fertilizer for them to grow in. That's not trivial and is probably half the work.
Right. So you're saying that this is a technology where we target the DNA of the plant. Plants are, of course, made up of millions of cells. And so you get this technology where you edit in some of those cells the DNA, and then you have to go from the single cell to a larger plant. And so that's what you're talking about, to regenerate that plant from an edited single cell. So from that perspective, does it always work?
Nearly always.
Okay.
If you've gone in with the knowledge base and understand where you want to make the single change that will have an effect, if you have been successful in making sure that the edit has taken place, that in some cases nothing happens, and if the change that you have made has not led to some change in the metabolism of the cell that means that the cell is less able to survive, then perhaps you've then been able to recover a plant. There's no reason why that plant at the end won't have the change that you originally set out to do. And based on the knowledge that you had at the start, you expected that you were going to be changing the color or changing the stature or changing the disease resistance of the plant. You could expect that to be the case. One of the handy things about plants as opposed to humans is that they always listen to their genes. They don't have free will. So if we do make a tiny change that is going to have that effect will be there to be seen.
Oh, that's interesting. I like that perspective of no free will for plants.
For plants.
So let's enumerate the things that we need to be able to gene edit a plant. Let's say I went into my garden, I had a favorite plant, and I wanted to, say, change its color. What are all the pieces of information and all the technologies that I need to be able to do that and get my plant gene edited to do what I want? Change it from, let's say, a red to a blue plant.
I'll talk about the technique in a second, but it occurs to me what I want to say there is that you also want to understand the really good reason why you want to do that. It's important to work out what the benefit is going to be. You're looking to make a change there that is not just for play. We tend not to do this kind of work just to understand or explore the edges of science. It's about trying to achieve an outcome that people want. So I encourage you to go into your garden and find where there's a need. And once you've got that need, you're going to need to understand some genetics and some molecular biology, some tissue culture, some plant husbandry. In terms of the genetics, you want to understand what it is that causes the characteristic that you want to change to be the way it is. Say it's the color of a tomato. You need to understand why the tomato is the color that it is. You need to understand what could make the tomato the color that it could be. It could take you 2 years, 5 years, 10 years of research to understand that. It depends on what 4 billion years of evolution has led to in terms of how that plant determines what color it is. Once you know that information, you probably need to look around and check that there's nothing else contributing as well. In lots of cases, an important characteristic of a plant could be determined by lots of different genes, all in chorus together. And if you're going to go in and make a single change, which is what this technology does, then that's like changing one person in a chorus. It doesn't really make the song very different. So you need to be lucky enough in your choice of target that a single change is going to make a noticeable difference. Once you've got that, you need to understand the DNA sequence. You need to find a place in that DNA sequence that's unique to the entire plant genome, all of the DNA that's in the cell. You want to hit it in one place, make one tiny change, then you need to find a place that's unique so that the scissors, which look for a unique site, can find and cut there. Then you need to be lucky, like we said before, and the change takes place. You don't get a change that corrects itself and becomes the ordinary, what we call the wild type. And you will have also had to solve the problem of getting your scissors your molecular scissors inside the cell to make the change. We do that in two ways, and that can be technically complicated too. So you're going to be spending a lot of time in your garden shed working on this plant. In some cases, we can introduce the molecular scissors into the plant cell directly. In other cases, we have to, in effect, introduce the instructions for making your own scissors into the plant cell. Those are both complicated, tricky. You may not get it right, and you'll have to keep working on that until you nail it. And then ultimately, when you make your change and you have what you think is going to be the colorful, or potentially future colorful tomato, you probably have a lot of other failures beside it as well, in the same flask, in the same bottle, in the same growth cabinet. You need to work out which one is the successful one. That's going to involve a lot of genotyping, a lot of PCR, a lot of measuring of what DNA is and isn't there. And then you're going to need to use a lot of subtle techniques of choosing media types and fertilizers and plant hormones in order to encourage that single cell to grow back into a plant. You've got a big job ahead of you.
Yeah, it sounds pretty complicated.
It's better to have a team. And like I say, it's better to have a few decades of accumulated knowledge of how plants work before you embark on this.
Okay, so once we've got a plant that's edited, you've regenerated it from the edited cell, you've got a whole plant, it's growing in its right medium, what's the next step?
Well, you need to do something that we've also been doing for years in our breeding programs, and that is some really robust evaluation of whether it's the thing you want. Not just because it's a plant that's got the single change. You believe you have that in hand and you've done some tests. But what you need to do is take that thing out into the field. Everything that we're trying to do with gene editing or with our other breeding programs is to try and make something that's better than what's already there. You want a better apple, you want a better potato, you want a better blackberry, you want a better this, that, and the rest. If it can't do as well as those, then it isn't interesting, even though you've made your little gene-edited change. In our breeding programs, we might produce as much as 10,000 plants a year, and the job is to remove the worst 9,999 of them. You've gone a long way through that process by making a gene-edited plant. You know you've got something that's pretty good, but it still needs to stand up against those others. The other 9,999 that we've produced, we're still going to throw those away, but they're already pretty good. We've been breeding these for decades. Your new plant with its gene editing, we need to know how it's going to grow out in the environment. We need to know how it's going to cope with the storms that it might meet, how it's going to cope with the climate of today and of tomorrow. We need to know that it's going to be productive. It might produce an amazing tomato, but only one. That's no good to us. We need to understand whether it's going to be appealing. We need to understand whether it's going to cope with being produced, harvested, shipped around the world, and enjoyed by a consumer somewhere. Until those evaluations have taken place, until we've understood whether it really is the best thing in our hands compared to the other 10,000 that my breeding program just made, then I'm afraid your gene editing effort won't get anywhere.
Right. But if you, let's say, picked a plant that was already from a breeding program and was already the epitome of fitness and whatever traits that you wanted to have, and then you gene edited it, would you still need to go through that process? Or is it because the editing process might affect your gene that you want to affect and improve a trait, but it might come at the cost of other traits?
We would absolutely have to assess that. You're dead right. You would have gone a long way to saving time if you gene edited something that was already pretty good, that was 99% perfect, nearly at the target that you want to achieve. That would mean that you know you're built on a really solid base of many years worth of making improvements. But you really have to test. We take nothing on trust in that regard. And so everything that's produced out of the breeding programs and out of any breeding program that was using gene editing in the future would still go through some kind of evaluation phase. You need to know that nurseries can propagate them. You need to know that growers can grow them. You need to know that the harvesting, packing, cool chain processes all work and that consumers are going to enjoy them at the end. And until you've proven that to yourself, you won't be putting it into the system. The future of breeding will involve gene editing, I would very strongly suspect, alongside and integrated with breeding. And that will make progress faster. That will make some of those early steps of where we have to produce 10,000 plants or more faster or more effective or more efficient or moving towards a target better. But that second half where we need to check what we've got and where we need to make sure that it's better than anything that's already there and that it hasn't got any new problems, we've still got to evaluate those every single time.
Right. Could we maybe speed it up by having, instead of 10,000 you mentioned, let's say, could we have 1 million plants planted out in an orchard and then assess them?
Yes, you could. Certainly had that challenge of what's the best way to make an impact through a breeding program. And breeding is often a numbers game. If you have a bigger breeding program than somebody else, you're probably going to be more successful. I wouldn't want to liken it to a lotto every single time. There's a lot of wisdom in choosing the particular tickets that we choose, that were going to be the winning ones. But nevertheless, there's a kind of random element. You can take out some of that random element if you're looking at, say there are 5 things that would make what you're producing a little more effective, and you could make 3 of those through gene edits. Then you're taking some of the element of having to have big numbers to be lucky enough to get the right ones in the right altogether in the right place. But I don't think there's a possibility that will entirely eliminate that necessity to still do the testing. Mm-hmm. But you might certainly be able to come up with a way in which you could very quickly evaluate much more than 10,000, and therefore you'd increase the power and likelihood of success. Our job is to get towards kind of end goals that have often been agreed with industry, or what consumers want, or what the future climate is going to bring for our growers here in Aotearoa. To know what those future targets are and to be able to build in the solutions or ways for our crops to cope with those as soon as possible would mean that we might not be doing 1 million a year, but maybe the 1 million that it takes to get there could be spread out over many years if we start work as soon as possible.
Right. What are some other opportunities for gene editing that would be important in selecting the plants that we're going to be gene editing?
I think the example that comes to people's minds is often something really visible, like are you going to change the color of something, make it more pleasant? But some of the largest bodies of research that we're putting effort into is about dealing with something that is less glamorous and shiny, but is probably the biggest crisis facing the age. It's facing agriculture, but it's also facing the whole of society, and that's climate change. Climate change isn't just things getting a little bit warmer. What that's going to do to fruit production, vegetable production, to the production of the food that we eat is going to destabilize it. And that's not just a concern if you're producing pretty nice tasting luxuries for people. That's about basic nutrition. So being able to support the growth of plants through changing climate is important. As the climate changes, not only does it get warmer, but it could be drier, it could be wetter, there could be more storms, there could be more diseases that didn't thrive before but do thrive under the new conditions. There could be new insects coming in that really enjoy the new warm version of New Zealand and really tuck into some of our crops. Those are challenges that we need to prepare the plants for. There's also, in terms of things like color and those sorts of characteristics, which people might think of as maybe more trivial kind of characteristics, what's important for New Zealand is being recognized for the quality of our produce. And whether we like it or not, people make a really snap judgment. Something that is appealing does count. Something that does really draw the eye makes them purchase something that's from New Zealand, and a benefit will flow back to New Zealand. So I wouldn't discount those sorts of traits. But ultimately, back here, our responsibility not only to our industries today who are trying to produce high quality crops, but also the generations that come next that are going to be living in that environment, is those generations don't want us to sling around a lot of chemicals. Those generations don't want us to use the land in ways that mean it's less usable further down the track. So that responsibility that we all have to our kids and our kids' kids needs to figure into the decisions that we're making in using this sort of technology. So we're also really interested in supporting the idea that sprays, for example, some of our industries here in New Zealand want to be spray-free by 2030 or 2040 or 2050. Ways to achieve that can be slow, can be challenging, can be really difficult. It involves farmers and growers and industry and all those who are working in food production to make lots of little changes. If we can take part in that, if back in the breeding program producing new varieties, we can introduce a single change with our molecular scissors that's going to help in that way, is going to mean that plant can be more robust and doesn't need the kind of protection that it gets from being sprayed to keep the insects off or to keep the fungi off, then we're adding to the achievement of that goal. And that's just as important, if not more important, as the other things that we're trying to take on. I'd like to think that we're covering all of the bases so that not just a few growers or a few people who work in the industry or a few people who care about overseas export dollars, but also the public thinks this is the sort of changes we need to see. These are the kind of varieties, cultivars, differences that we hope are going to be part of New Zealand's future, so that New Zealand's future is a better future, so that our soils are great, so that our water is clean, so that our farms are sustainable, so that we're still successful, but we're also clean and green.
So in terms of once you get these plants, you've got the gene-edited plants, you've tested them for all of these traits, is it then just good to go? Or is there more before the consumer can then start buying whatever crop that this plant is producing?
Here in New Zealand, you couldn't, at the moment, take that work that you've done, take that outside and plant that outside, evaluate that new plant against the old ones, grow it up at large scale and sell it to consumers. There are regulations in place to prevent you from doing that. Those regulations were put together in order to make sure that there's a safety assessment and an understanding of the impacts beyond what fruit or vegetables are produced, what the social, economic counter impacts, what the positives and negatives might be. And they weren't put in place for this technology that we're talking about. But those regulations are being reconsidered and re-looked at now. And from our point of view, we think that there's a real benefit to people understanding what's going on, and where there are regulations to be applied, then you apply them. Where there's an understanding that there is be a good opportunity here and not the risk to bring things into the outside world, and that's something that people can agree on how that would work. So in that situation, perhaps you could immediately get out there into the world, but that's not the world that we're in there right now.
Right.
There's still change going on, and that kind of change is going on all around the world as well. Different countries are at different speeds in different places in coming together on whether they harmonize or align or have kind of rules governing the release of these sorts of plants into the environment. They're not all the same. The rules are not implemented in laws to the same degree in different countries. So we're undergoing a period of change in this. The opportunity is clear from the science point of view. The opportunity is clear in terms of what can be achieved in delivering targets that are really useful to our industries. But there are still regulatory issues and political issues and social issues that are important conversations that need to take place as well.
Yeah, it sounds like it would take a long time to sort of get through all of that. So obviously on one side is gene editing speeding up the process of selection and breeding. And the other side is the, sort of the regulatory considerations. How long would this whole process sort of take?
Looking in terms of the sort of rules that are being implemented in countries around the world and which are being considered for here in Aotearoa, you would still have several years worth of analysis and regulatory understanding of what's going on in terms of your experiment to deliver it out into the world. Ultimately, there's a view to say that we would be able to understand sufficiently what's going on, that you could treat plants that have been edited in very small, precise ways as being just like plants that have emerged from a breeding program, because the changes are the same. There isn't anything different except the amount of intention behind the change that was made. The actual change that is made is something that can arise naturally, or it can arise through the clever use of molecular scissors. In that case, there's no reason to treat those differently. In other cases, there are versions of this technology where larger changes could be introduced, where new pieces of DNA could end up in the genome, deliberately put there in order to create an effect, to get to the endpoint that's being sought. Those still raise for some people questions about whether that's too large a change to just be put out there into the world, and therefore there needs to be regulatory oversight, more broader understanding, and a kind of license to do that. And that's a set of regulations that would be more nuanced than we currently have, that would be looking at where there is a degree of change that people can be comfortable with, as comfortable as they are with the outputs of a breeding program, then you treat those two the same. And where there's a difference that people have different thoughts about, then you treat them differently. Perhaps that's something that allows more people to be engaged in the conversation and to understand what's really going on in breeding programs and in trying to deliver what we're trying to deliver. And it would be great for people to be more involved in that and understand just what it is we're trying to achieve.
Right. So obviously the regulatory hurdle remains. That might take X number of years, who knows how long. And I suppose we'll find out soon enough. But once you get past that hurdle, how quickly are we talking to deployment, seeing things in stores? Are we saying like a couple of years? Are we saying a couple of decades?
I think we're quite likely here in New Zealand to see things in stores from offshore before we see them out of our work here. Certainly work that we've already begun in containment, in controlled glass houses, in our laboratories. You could see that if the regulations permitted and if what was produced was exciting enough and was meeting the targets, then you could see those within a couple of years starting to emerge. But there is still that process of evaluation and of scaling up to a size that makes a difference to an industry. So still 2, 3, 4, 5 years before these things are really coming from our work out into the world. However, offshore, in other countries, they're already moving fast. And just like any other technology that affects agriculture, we see that being deployed at scale all around the world and then imported into here. The way in which material arrives in New Zealand, in terms of imports of processed food or fresh foods, is a very different process to what happens when something starts in a laboratory here, goes through a breeding program and emerges out into orchards and fields in New Zealand. It's a much faster process. So I think we'll be seeing them from offshore before we see them onshore. We really want to catch up. There's a lot of benefit to be had from these, but we're starting yesterday, today and tomorrow, whereas other people started 5 years ago, 4 years ago, 3 years ago. We need to move fast.
Yeah, I think it's such an obvious thing that you said, knowing the way that the world works now. It's a globalized world. And with that sort of level of interconnectedness, it's a major threat, to our local industries if we don't catch up. I definitely agree that that's something that we're under pressure to change. But of course, as you said, it's important to be careful about these and be very methodical and deliberate about how we assess these products and that sort of thing moving forward. I'm obviously a big fan. Do you ever think that there'll be the day where you can sort of design from scratch a plant variety with, I don't know, let's say 10 different traits, all gene edited to get the ultimate fruit crop? I don't know what that looks like, but do you think that would be possible?
I think that day is coming. I think it's a ways off yet, not outside the realms of possibility, but I think it was important what you said there about you're not quite sure what that looks like. That's sort of the problem, in that today what we think of might be the perfect fruit or vegetable or any other crop that we might want to make changes through breeding, through gene editing towards. That opinion's going to change. That opinion's going to change because of how climate is affecting our crops. It's going to change because consumers want something different, something healthier, something more exciting, something more pleasant, something that tastes better, something that they know has been produced with no chemicals, those kinds of things. As those needs change, those wants change, then the definition of what the perfect crop that we might be able to create with 10 or 12 or 20 or 40 different changes in it, that's going to change too. So there'll always be a place for providing new cultivars, new varieties that can meet those new needs, but we wouldn't want to get too far ahead and predict what the perfect opportunity is in 2080, because we've got things we need to do for 2030 and 2040 and 2050, and those are the job in hand.
Yeah, it's an exciting time. I'm really looking forward to it. And obviously it's still a while before we have, CRISPR products in our CRISPRs, let's say. So in the meantime, you look over a large team of scientists that are motivated, driving towards this greater outcome of gene-edited crops. How do you motivate people? What do you tell them to have an eye on the future?
I think everybody working in this area isn't getting out of bed in the morning so that they can explore what DNA does. They're all here because they've signed on to making a kind of difference in the world. Mm-hmm. So science is a vocation. People are self-motivated. It's not really about stirring people and getting them to be at all interested in this. They're interested. It's about aligning. It's about understanding just what might be the very best change in the world that we could do. And do we have the tools to be able to do that? And do we have the people at kind of the end of the line who are saying, yeah, we want that? If we have those things in place, then all of those scientists will leap out of bed early in the morning and get cracking on the job, which is great. Nobody needs to be pushed into doing this sort of thing. We all just need to be able to look towards a horizon and agree what the point on the horizon we're going towards is, and then we find ourselves running towards it.
Oh, absolutely. I totally agree. That's an amazing view of the future. Well, thank you, Zac, for coming in and having this chat with us.
Thanks, Jay. Really enjoyed.
And thank you, listeners, for joining us for this Scigest episode. Please rate and review us on your favorite podcast platform. And if you really like what you heard today, please tell a friend. Word of mouth helps us grow in the best way possible. Thank you.